And welcome back. I trust that many of you were here last week for the first installment on photosynthesis. Our speaker, Marshall Elzinga, is a graduate of Hope College, earned his degrees in biology and chemistry. Then he went on to the University of Illinois in Urbana, where the university awarded him a doctoral degree for his work on the differentiation of hemoglobin in the amphibian Rana catisbiana, known to the rest of us as the American bullfrog. Marshall will gladly answer all your questions about red blood cells in the bullfrog after this presentation. Marshall was gainfully employed by the Brookhaven National Laboratory in Long Island, an institution that has a very long and rich history of scientific discovery. He then went up the coast to Boston, Cambridge actually, where he worked at the Harvard Medical School, which was founded in 1782, long before Marshall was there. But we're here today to enjoy the second and final installment on photosynthesis. And today, Marshall will focus our attention on the mechanism of photosynthesis and also on the cellular metabolism for energy. Without further ado, Marshall Elzinga.
Well, yes, well, welcome back. I think you mentioned that last week as well. Yes, I used to be a trombone player in various community orchestras, including the Holland Symphony. In fact, I met my girlfriend here, who's a guest today. She was a bass player in the Hope College Orchestra back in the late 50s, 1950s. And that was the beginning of, what, 63 years now? Being married. Anyway, she's put up with me for these years, and occasionally we talk about things, although I like to point out that the only thing we have in common is music. The only thing we have in common is music. Anyway, the talk last week was on some of the history of photosynthesis. And as I said then, this is not really my major field of interest. I worked on hemoglobin of bullfrogs back in the 60s to get a PhD. And then at Brookhaven Lab, I worked on the structure of something called carboxypeptidase B. It's a digestive enzyme made by the pancreas that helps in digesting protein molecules. And then that was as a postdoc, and then I moved to Boston and worked on muscle proteins. And so we'll talk a little bit about that later, but now we need to talk about photosynthesis again. I've learned a lot over these past few months boning up for this. Photosynthesis is an extremely important process. And so I will try to brag about or emphasize how important it is to life on Earth. In any case, let's review a little bit of the stuff that we talked about last time. The reason I have you on this side is because you fit on this side, number one. Number two, this pointer only goes to one screen at a time. So last week I was sort of going back and forth, but I think it's more convenient this way. Apparently online it does not, the pointer does not show, but they have to live with that. Anyway, the process we're going to talk about is photosynthesis. Just to remind you that plants take sunlight and carbon dioxide and water and churn out, so they throw away oxygen, which we then use, and they make sugar. And this is, again, a summary of things, but I'll go into that much later in the talk. This is a wildflower garden at my farm. We talked a little bit about my farm. I used to take half people to the farm in the summer. It was fun. Maybe we should do that again. But anyway, this is a barn built in 1892 by my grandfather. This is a wildflower garden that I planted a couple of years ago. And it's doing a lot of photosynthesis, not right now, but it will this summer to generate some nice colors. A brief review and then a week and a look forward. Last week we talked about a little bit about the origin of life, but the key ones are isogenic biosynthesis, which means it's photosynthesis without producing oxygen, and then oxygenic photosynthesis, which is really the star here. A little bit about plants and animals. Plants, of course, do photosynthesis. Animals benefit from photosynthesis. So today I'm going to talk about how photosynthesis works. How does respiration work? Because respiration is how we use the product of photosynthesis. How do plants and animals use ATP? ATP is an absolutely crucial part of this whole story, and I will try to talk a little bit about that. Glucose, starch, and cellulose, and then finally oil, gas, and coal. I don't know much about that, and it's not really a difficult thing to talk about because the products of photosynthesis have turned into oil, gas, and coal, which we all use. Anyway, the origin of life. When? Probably 3.8 billion years ago. That's a few hundred million years after the Earth was formed. When we talk about these years, we talk about, a year or two, 10, 20 years, or we're maybe average 70 years. It's such a small length of time compared with how long life has been on Earth. But that's what it is. The requirement for the origin of life, well, we don't know how life originates, and we probably never will. But the requirements, we think, intense sunlight plus whatever hydrocarbons were around. Hydrocarbons are food, and some of these were made naturally using hydrogen, the effect of sunlight on hydrogen and carbon dioxide. Was life improbable? Yes, but the raw materials were present. There was lots of time, and it did happen. Could it happen again? Probably not. We now have too much oxygen around. We depend on the oxygen, but oxygen has a lot of negative effects on things that can't use it, and so the chances of life originating again are very small. The evolution of photosynthesis, anosogenic, in other words, photosynthesis that does not produce oxygen, goes back maybe 3.5 billion years ago, made ATP and glucose, but did not release oxygen. The key event required for anosogenic photosynthesis, the evolution of chlorophyll and a reaction center, and I'll talk about reaction centers, not too much about chlorophyll. Oxygenic, this was the key event that made possible higher, plants and animals, maybe a billion years later. So that, again, we're talking huge lengths of time. But that oxygenic photosynthesis makes ATP and glucose, plus it releases oxygen. And oxygen is really a throwaway product for oxygenic photosynthesis. Oxygenic photosynthesis changed everything irreversibly. The availability of oxygen made it possible for plants and animals to extract large amounts of energy from glucose. In other words, glucose was being made, but it really requires oxygen to extract glucose for our use. Cyanobacteria, the big stars here, these are bacteria that could do photosynthesis. We think oxygen photosynthesis evolved in something which is a precursor to cyanobacteria, that is bacteria that were more primitive, if you wish. But cyanobacteria put it all together. They could make, they could use sunlight to make glucose and ATP and release oxygen. Cyanobacteria have been responsible for building up oxygen levels in both the oceans and the atmosphere. In the oceans initially, because that's where life originated, and that's where living things live. And so the buildup of oxygen levels in the oceans had a lot of effects. There was no oxygen, very little oxygen in the atmosphere early on, partly because any oxygen that was produced would react with iron that was in the oceans. And we call that the rusting of the oceans. That stuff settled once there was enough oxygen to rust, to react with all the iron that was in the oceans. Then oxygen levels in the atmosphere built up, and that made possible life on land. Endosymbiosis, I talked a little bit about that last week. I don't really want to, it's a complicated concept, and I don't really want to go into it. It simply points out that mitochondria are in our cells. I'll talk about mitochondria a bit. And the way they got there is probably by having a fairly large bacterium swallow a small bacterium, and then that small bacterium eventually became a mitochondria. And of course, mitochondria now are in everything that's living. And that chloroplasts in some of these early tiny bacteria were eaten by a larger plant. And plants and cells that swallowed both mitochondria and chloroplasts became plants. Those that swallowed only mitochondria and not chloroplasts became animals. And that's why we are not green. We animals cannot photosynthesize. And again, it's speculative, the concept. We generally think it's correct, but it would be almost impossible to test. So finally, photosynthesis. How does it work? Well, there are two distinct steps in photosynthesis. The first step makes ATP, and that requires sunlight. The second step makes glucose, and that can happen in either light or dark. So we'll talk about a light reaction and a dark reaction, but the dark reaction isn't really exclusively dark. It can be take place in both light and dark. And now we're going to get into some pictures that some of you will immediately recognize. You chemists will recognize as hey, we learned all about this before. Others will be sort of blown over by the complexity of biological molecules. But I'm just going to try to explain what they do, not all the chemistry of it, just some of the basic chemistry. ATP, adenosine triphosphate. First of all, I should, this is it right here. This is something called adenine. When you add this sugar, ribose, it becomes adenosine. And then when you add the phosphate groups onto this sugar, it's adenosine triphosphate. This you'll recognize as part of DNA or RNA. It's a widely used molecule by living systems. And it's magic. I mean, it's just an amazing molecule. So what does it do? Well, I'll talk about this more later, but the fact is that this bond here between this phosphate, we call the beta phosphate, and this one called the gamma phosphate, that bond is the one that holds energy in biological systems. I'm going to talk about it later as sort of a currency of life, or the money of life, or a rechargeable battery. There are various ways we describe ATP, but it can accept this phosphate, which now contains energy that we can in fact use. There's a cycle to it. ATP, when it loses its phosphate, when it does cellular work or biosynthesis, for example. Muscle activity requires that phosphate to be released. Building things within the cell requires that phosphate to be released. It then becomes ADP, adenosine diphosphate. There are only two left. And then that gets recycled back to ATP. And I'm going to talk about how that happens. Glycolysis, a breakdown, partial breakdown of sugar, The TCA cycle, which is mind-bogglingly complicated. Oxidative phosphorylation, the same. But for our purposes today, photosynthesis. Photosynthesis takes ADP, turns it back into ATP. So when we talk about ATP synthesis, we're not talking about making it de novo. We're talking about taking ADP and turning it into ATP because this has the energy. And glucose, this is the other molecule we'll talk a lot about, ATP and glucose. Glucose is this molecule, this long molecule here. It can exist in an open form or a closed form, circular form. I talked about this last week. It has 6 carbon molecules, 6 oxygens, 12 hydrogens. The glucose molecule that plants make can be either linear or circular. And it's in the D form, not the L form. And that refers to the ability to rotate light, but it really reflects the position of this oxygen here. If this oxygen, this is a hydroxyl group, if that's on this side, it's L, levo, meaning left, it rotates it left. And we cannot use that, but in this position we can. It also has an alpha form or a beta form that really refers to the form it assumes when it combines with another molecule, and I'll get into that. But that again refers to the position of this hydroxyl group. If it's down, it's an alpha. If it's up, it's a beta. And these are really just paper constructs, just so that we can talk about it. In real life, they don't look exactly like that. Anyway, the alpha form can become starch. The beta form can become cellulose. And again, I'll talk about that. At this point, let me introduce any questions so far? All right, we'll just keep going here. A typical plant cell, everything happens within cells in living systems. I mean, we have a few trillion cells. I mean, we're just made up of a bunch of cells put together. It's like a Lego set that has just everything put together in the right place. Okay, so here's a plant cell and a bunch of plants, some of our favorite entities. And this would be a plant cell from a leaf. It has a lot of stuff in it. Golgi vesicles, the ribosomes are responsible for protein synthesis. A nucleus, that's where its DNA is. Central vacuole, that's unique to plants. We don't have those things. Cytoskeleton, microfilaments, microtubules, and so on. Membrane. Mitochondria, these things here, each cell has maybe 20 to some nerve cells have 2,000 mitochondria. There's cytoplasm, that's sort of a viscous liquid that surrounds everything. And it has chloroplasts. A typical plant cell will have quite a few, but this shows just 3 of them. You keep going on this water, it's good water you have back there. Okay, so the chloroplast, we blow it up here to just emphasize the things that are in it. And this is the chloroplast again, and this is a blow up of a structure of a chloroplast. It's an idealized structure, but it basically has a cell outer membrane surrounding it, and it has stuff inside. Now, this is the structure that we think actually got into the cells because it was in a small bacterium, and it was eaten by a larger bacterium, and it eventually got settled in there. And one of the evidences for that is that that chloroplasts and mitochondria have their own DNA. So that we think of that is still remaining from the original thing. It doesn't code for all the things in a chloroplast or a mitochondrion, so it's a mixture of things. But it works. It goes back billions of years. I mean, that structure what that we see is an extremely old protein molecule, and I'll talk about that. Anyway, this is the way they're sort of stacked up. These things are called, this is called a granum. It's a stack of thylakoids. I don't know where they get these names, but that little disc is a thylakoid. Blowing this chloroplast structure up some more. Well, this is actually an electron, a microscopic picture of a plant cell, of a leaf. I don't know what leaf that's from. But anyway, it shows that there are, in fact, these chloroplasts. They're little green things in these cells. This is blown up. The chloroplasts are scattered around. So I'll show you mostly diagrams, but these things actually exist as cells. You can look at. So the tools for making ATP, and that's what we're talking about. Remember, with the first step of photosynthesis is to make ATP, or to actually transform ADP to ADP. They're in these thylakoids, in these yes, well, in the chloroplast. Here, this is shown diagrammatically as the DNA of the thylakoid, of the chloroplast, but it's just loaded with these things. And the thing that we're going to focus on now is this little disc that has a lumen. That is, there's some fluid inside, and what's important is the membrane surrounding the thylakoid membrane. So much of biology is done on membranes. The nervous system operates by having holes in axons of neurons or the cell body, so that ions can pass through. That controls the electrical activity of your brain, and I'll talk about that a bit later. But almost everything where that something is done is in membrane. Okay, so here again is that idealized chloroplast. Here's the lumen, the membranes, and these are the things that sit in the membranes. Now I've got these lines. This is the membrane with the yellow on one side, on both sides. It's actually made up of these molecules that have a lipid inside and a hydrophilic outside. But things get stuck in these membranes, and that's where things work. A picture like this doesn't exist within a cell. I mean, these things are all these things are all together. These four things work together as a unit. So we really don't know how they're distributed within the cell, but there are thousands of these things, these sort of functional units within the membrane of a given thylakoid. So the leaf is fairly translucent, so that the light can come through the outside of the leaf into the interior of the cell, and impinge upon these structures. And the two that are important for light are this one here, which is called photosystem 1. I'm sorry, photosystem 2, and this is photosystem 1. The reason they're named that way is because that's the order in which they were discovered or described. They actually function by having some things go from photosystem 2 to photosystem 1. So the light impinges on both of these, but let's look at this one first. The light hits this, and there's a reactive center within this light. And the important thing is this thing down here. It doesn't look like that. It's just there because things happen there. And the event, which is probably the most important in all of life, is a reaction center here that breaks water into hydrogens and oxygens. That's called the oxygen evolving complex. And I'll show some pictures of that. They won't really mean much to us, but they do. There is an understanding of these, and it's extremely important. Anyway, it breaks the water molecule into these protons. Hydrogens are single hydrogens, proton, and oxygen. Now, it shows just one, but well, it shows one molecule, but in fact, you end up with a free oxygen. Oxygens don't like to be alone, so the oxygens join up, so you have two. So you have O2. That's the way oxygen is in the atmosphere. Anyway, this is just thrown away. It is not used by photosynthesis itself. What photosynthesis requires are these protons. And the evolution of the reaction center for that is considered one of the most important events in the history of life on Earth. Anyway, the protons, these protons migrate over in this direction. They sit in the thylakoid lumen, and we need a lot of these in the lumen. I'll explain why in just a minute. The other thing is that there are also electrons that are released. They go up into the photosystem, and this is really mysterious stuff. I mean, I worked at Brookhaven next door to a guy who was studying this, and they use very fancy machines, but you can't see electrons move. We just know that they move from one place to the other and eventually cause things to happen. So these electrons pass from this photosystem 1 down through something called plastoquinone, through cytochrome, eventually to this protein, and then to the photosystem 2. And as these electrons pass, they somehow cause these protons that are on this side now, the stroma side, to pass through the membrane and accumulate here. So not only does the oxygen evolving center release protons, but also protons come down from here. And these electrons get here, and then this photosystem 1 also receives light. It apparently gives them a little kick. It activates these electrons because they've lost some energy from going from here to here, and goes up into this section here, and that produces NADPH. Now, I really don't want to talk much about that. It's nicotinamide adenine dinucleotide phosphate, and it's a well-known molecule, and it helps to make glucose. But it's enough for us to deal with these protons going over here. So these protons now accumulate here, and they are reduced in number here, and they then, and there are more of them down here in the lumen. So they end up here by this molecule, which is one of my favorite molecules beside the ones that I worked on. And the hydrogens actually accumulate here. There's a channel that goes through this thing. It's sort of a tube. And once they reach the top here, they cause this top to rotate. And that rotation can actually be seen. I can explain later if you wish, but this rotates, and as it rotates, it takes the ADP and converts it to ATP. There are some theories about exactly how this works. Enzymes catalyzing reactions basically have it's a lock and key mechanism. These small molecules enter the surface of the enzyme, which functions as a catalyst, and it helps it to change into something else. And so in this case, the energy really is from the osmotic energy of the high number of protons here passing through, and it causes this to rotate, and that gives us ATP. This looks at it a little bit higher, a little bit larger, and again, it's still a fictional. It's a cartoon, if you wish. It has all these parts: light, photosystem 1, photosystem 2. Here, the water being broken. Water is an extremely stable molecule. We require huge amounts of energy, of electricity, to break it if we want to make it into to hydrogen, so that we can use for driving cars or using other methods to generate power. So this is really the only thing in nature that will split a water. Anyway, that shows then the hydrogens coming across, the ATP synthase, this hole that goes through here, and the ADP turning into ATP. Within the photosystem too, there's this water splitting site. Now it's these things, these that's aspartic acid, glutamic acid, these are all side chains of the protein molecule in which this water splitting site sits. And the water splitting site itself has 4 atoms of manganese, 4, 3, 2, and 1, and an atom of calcium. It exists nowhere else in nature. It's been solved by X-ray crystallography, and some people who work on hydrogen generation are hoping that they can eventually make this in the laboratory and use it for splitting of water and generating hydrogen. It has not been done yet, but that is one of the hopes of this whole thing. So that's that sits down in here somewhere. I should know, but I don't know exactly where. But anyway, that's where ATP synthesis takes place in the light cycle of photosynthesis. A closer look at this ATP synthase, and it's kind of an interesting story. I have some time here to celebrate a 50-year anniversary, actually. In almost exactly 50 years ago, this is in March, late March of 1973, we were living in Germany, in Heidelberg, Germany. I was basically on a sabbatical leave, where the American Heart Association was supporting my research, and they encouraged those of us, it's called established investigators, to take a year off somewhere and learn something that you want to learn. So I went to Heidelberg with the whole family, 3 boys and my wife, and we had a great time there. But in March, I had just published some papers that many people were interested in, and so I was invited to give a lecture in Paris, University of Paris, the medical school there. And I did not take Jackie with me. I took by train, went over to Heidelberg first to Dijon and then to Paris to give these talks. Sort of many of the students attended, and they didn't understand English, so I had sort of a simultaneous sequential translation of the talk. But anyway, I was talking about actin and myosin, which I'll talk about a little bit later. But there was a man there named John Walker who I did not know, but he was at the lecture because he was interested in the muscle proteins, because he was looking for a project to do. So I got to talking with him. He was not a friend. He was an Englishman. He had done his PhD at Oxford and had done a postdoc at the University of Wisconsin, looking for a project to work on. He was a very bright guy, obviously, and he was looking for a project. So he had spent 3 years in Wisconsin. Then he came to Paris at the Pasteur Institute to work for 3 years, again, still looking for a project to work on. And he came to my lecture because he was interested in muscle and perhaps working on muscle. So we got to chatting, and he took me over to the Pasteur Institute, gave me a tour of the Pasteur Institute. And then I didn't keep in contact with him. A couple of years later, he and I were at a conference in Cambridge, sort of running this together on protein structure. And then I didn't hear from him again until 1997. That was in the early 1997. Here's John Walker won the Nobel Prize. And so the Nobel Prize was for work on this structure here. He had moved from Paris then to Cambridge, to one of the finest laboratories in the world, the Molecular Biology Institute at Cambridge. And that's where crystallographers got their start. The first structures were solved by Perutz and Kendrew, and hemoglobin and myoglobin. Anyway, he worked on this and was publishing the structure. And so the structure that he ends up with is, well, first of all, it's an enormously large protein to do structure on. And so electron micrographs make it look like this. So the picture looks like this. The actual microscopic picture looks like this. It's a special electron microscopy. And the structure that you end up with something that looks like this. And these are protein chains, all of these things that look like a coil, that's an alpha helix, and other things are non-alpha helices. Anyway, there's a tube that goes through here where the protons go through, and this is where this is the part that rotates to make ADP to ATP. And it's very difficult to do this kind of stuff. I actually was spending the time in Heidelberg working with a group doing X-ray crystallography on a small protein, about 1/20th the size of this. And we got a paper on that. But it remains very difficult to do this. But at this point, it is possible. And the techniques have improved tremendously over the last 40 years. And so now we have maybe 1,000 that have been solved. But I was reading the New York Times. I always read it first thing in the morning, and there's a column by Tom Friedman. And Tom Friedman is one of my favorite writers. He writes about everything in sight, lately about his distaste for Benjamin Netanyahu. Today he was talking about artificial intelligence. And he had talked with somebody from Google who was describing their version of artificial intelligence and showing him how they could give the machine an idea, and he would generate 200 words on something, 1,000 words on something, a poem about something, whatever you want. But he pointed out that what it's being used for now is prediction of protein structures. Now that's always been a goal of people who work on proteins, because proteins are very, very, very complicated. They're organized. They're chains of amino acids that fold up in a particular way, and they have to be folded up just right to function. So how do you figure out how they're folded? Well, you do X-ray crystallography, but people have always been trying to predict them. And so now this person thinks that, and they've experimented. I mean, you can do a test. You can take a protein sequence, give it to the machine, and see what it predicts. And then you can compare it with what it actually looks like, because you've done the experiments, and it's apparently very, very good. Scary. Anyway, also, well, I don't know if you have any questions. We can go back to this later if you have questions about it, but it's just a remarkable molecule. Again, it's been around as long as photosynthesis has been around, because you must have a way to take the protons made by release of protons. You must have a way to use those to generate ATP, and that then goes back to the origin of life. You have a question? Yes.
Question for people online. Was it Walker and his group who determined these 17 structures that are assembled to make ATP synthase?
Question is whether Walker and his group did all of this stuff. The answer is no. There were a lot of people working on this, different parts were solved by different people. It's just an enormous thing. So yes, no, many people worked on this, and he apparently was considered to be. He shared it with a person who had a man named Paul Boyer from UCLA, who had predicted some of the activities here. Predicted that there was a unit of that there was a circular thing that made ATP. So he shared it with. But other crystallographers certainly contributed. It's always the case. The Nobel Prize. Okay, so besides making ATP, there are the two electrons taken from water by photosystem 2 are activated by light harvested by photosystem 1. I just indicated that. They get reactivated, and they go up here and make this. This is going to show up in subsequent slides. So you need them both to make glucose. So this is the light system. The Calvin cycle is the dark. It used to be called the dark system, but in fact works in both light and dark. And it basically takes the, it makes glucose. Here's your glucose again. And this is the Calvin cycle. And it's a series of reactions that take place in the chloroplast stroma, in sort of the cytoplasm of the chloroplast. And it's not really a circle. I mean, it's a series of reactions that take place that have to be sequential, so they can be drawn as a circle. Makes sense to do that, but it's not really the way it works in the cell. Anyway, I'm not going to bore you with all the details here. It's very tough stuff, and those of you who have degrees in either biochemistry or medicine were required to learn this stuff at some point. But anyway, they take place in the dark or the light. It incorporates or fixes carbon dioxide. Okay, we know that carbon dioxide comes out of the air, ends up in the glucose. Well, this is where it happens. It gets the energy from ATP and from NADPH, and it produces glucose. So it's a cycle, so you can start really anywhere you want to. But let's start right here, where the carbon dioxide comes from the air and goes into this cycle. It goes in by way of something called rubisco, which is an enzyme, huge enzyme. It's considered to be the most abundant enzyme on Earth. And it incorporates the glucose, this carbon, into this five-membered thing in the cycle before, ribulose 1,5-bisphosphate. Somehow this carbon gets incorporated into this to make it a six-membered molecule. This then is split in half into two threes called 3-phosphoglycerate. Then by using ATP, we convert that into this one here. This is where we use some of the ATP. This is an energy requiring reaction. In other words, there's an enzyme involved, and it takes ATP, breaks ATP, and it helps the enzyme to incorporate the energy released into this thing here. The next step requires this NADPH, converting 1,3-bisphosphate glycerate into glyceraldehyde 3-phosphate, this thing here. And part of these are split off then and become glucose. So in this part of the thing, we take carbon dioxide, we use ATP, we use this, and we end up with something that can become glucose. Then we keep on going to other molecules. We need some more ATP here to make this, and then we'll go down the circle again. And so it churns out, it takes glucose, it takes carbon dioxide, flips out glucose, using the energy from ATP and NADPH. That's all I'm going to tell you about making glucose. It's the work of thousands of people. Many people made small contributions, but this is how it works. Yes?
And they named the university in yeah, that's what
what happened to my slide here? I've got a slide that it's supposed to say. Did I take that out? It says Calvin is Melvin Calvin, not John Calvin. It must be back here a little ways. Where is it? Yeah, here it is. Yeah, Calvin. Melvin Calvin was at University of California. just being a wise guy, right? The question was whether the John Calvin is the same as the Calvin for whom Calvin University was named, and the answer is no. Okay, so there we made glucose. Okay, so yeah, this is sort of a we've talked about how photosynthesis uses energy from the sun to make ATP, releases oxygen, dumps it into the air, uses ATP to make glucose, the Calvin cycle. Now, I think it's, I didn't want to leave it here. My choices were to try to explain that in detail, and I thought you would all glaze over. It's tough stuff. But I will try to explain how we actually use glucose and oxygen, because the benefit of having photosynthesis is so that things that can't do photosynthesis can use the energy from the glucose. So extracting energy from glucose, respiration. So living things extract energy from glucose to make ATP. I mean, that's just the opposite of what we did before. We used energy from photosynthesis to use the energy to make glucose, to make glucose and ATP. So now that we're doing just the opposite. In fact, the Calvin cycle can be seen as sort of the opposite of the reciprocal of respiration, but it's not really. They use different enzymes and so on. So the extraction of energy is called respiration. That's the process by which we extract energy from food. Respiration requires oxygen. That's why we breathe. Produces ATP. I'll explain what we use ATP for. And it occurs in our mitochondria. Most cells contain between 220 and 2,000 mitochondria. The large number is for long cells like neurons that run from your brain to your legs. And they'll have lots of mitochondria scattered along because they need to produce ATP all along. Anyway, so here's our plant cell again. Here's a mitochondria with some of the structural features. Here's an actual picture of a mitochondrion, a microscopic picture, showing this outside membrane, and then these cristae, these invaginations throughout, and that's really where things take place. Again, I said most of these reactions take place in membranes. Here the membranes are in the insides of mitochondria. So it's very complicated. Again, even more steps than in the synthesis of glucose. There are just a couple of things I want to point out here. First of all, each of these steps is mediated by an enzyme. We have thousands of enzymes. Again, these are proteins. They function as catalysts to facilitate a reaction. Sometimes the reaction requires energy because the products have higher energy than the reactants, and so that requires then help from an ATP. Some of them don't, so they don't require. They just facilitate a reaction from a higher energy to a lower energy. Part of this happens in the cytoplasm, and part of this is again a cycle in the membranes of the mitochondria. So in the cytoplasm we have glucose. That's where we start. Remember, we make glucose by photosynthesis, and we convert it to glucose 6-phosphate that requires ATP, requires energy. That's the only enzyme I'll actually name because some of you may remember that's hexokinase. This gets converted to fructose 6-phosphate. It's just a rearrangement of molecules between glucose and fructose. They're very similar. ATP then is required to make glyceraldehyde 3-phosphate. NAD is required to make this one. Then this one generates electricity, generates ATP, where 2 ADP results in ATP. And then this one puts out a water. And then we get to here. And then we go from here to pyruvate. And that again gives us 2 more ATPs. So this reaction here actually extracts some energy from the glucose to give us more ATPs than it needs to operate. So we start, this part requires 2 ATPs, minus 2 ATPs. This part is a net plus. So we actually generate a little bit of ATP. The real work is done here in this cycle. The pyruvate goes into this cycle. We have citrate. Then this requires energy from this reaction to give us alpha ketoglutarate, or ketoglutarate, and then this is actually here we flip out 1 CO2, 1 carbon dioxide, and then this one flips out another carbon dioxide. So we're getting rid of some carbon dioxide, and that's why we breathe. We exhale carbon dioxide. This is the carbon dioxide that we're exhaling. It's in this cycle, and it is being thrown away. And then we come around here to this. We come around again, and we just keep going around, extracting energy from ATP, getting rid of carbon dioxide. I want to look a little bit closer at this part here because this happens within the mitochondria. I think the next picture shows it even more higher. So this is the electron transport chain. It's something like what we saw in photosynthesis. The electrons flow through this and get activated in the complex. But anyway, we generate protons. Now in photosynthesis, we generated protons by breaking water. Here we generate protons from the glucose itself. It doesn't require as much energy to do that. As I said, the extraction of energy from water is very unique, very difficult, and happens only once, only in photosynthesis. Here we generate these protons, and these protons will go through this ATP synthase. Now that's the same ATP synthase that we saw in plants. ATP synthase is so unique. It's an enormous molecule. It's complicated, and we think it evolved once, and it's been around for all these couple billion years. It's changed a little bit because we have mutations and so on, but in function and in shape, size, function, it looks very similar to the ATPase that's in, for example, cyanobacteria, in the molecules that cyanobacteria have to make ATP. So here we go. These protons go through here, and we take ADP plus a phosphate and make ATP. Where do we use the oxygen? We've talked about using glucose as the energy source. In that cycle, we throw away the carbon dioxide. What about oxygen? Well, oxygen is used down here. When these electrons are released, they pass through here, and they accumulate. They float back into this stroma. Again, this is all idealized. I mean, these things happen, but we don't know that they happen in a picture like this. But anyway, the functional role of oxygen then is to react with leftover protons that come through and the electrons that come through here. It basically is sopping up the stuff that was used to make the ATP. It's a garbage collector. I mean, oxygen should really have a more sexy usefulness, but it doesn't. So we breathe to get oxygen to do this, and we exhale to get rid of the carbon dioxide that I showed in the previous one. It even says here, CO2 is a waste product. So just a bare bones summary, this really isn't very good, although it does simply point out that we start with glucose. We get some ATP by processing it in the cytoplasm, in the cytosol. Then it goes into this cyclic system, and substrate level, I won't complicate things with explaining this. But anyway, this whole system in the mitochondria produces a lot of ATP. We get 2 here, and 34 here. These are sort of calculated numbers. But anyway, that's where, and that's how we get energy from the glucose. We made it in it by photosynthesis. We extract it in our mitochondria. Now, how do we use it? Well, ATP is what we call nature's currency, and it's kind of fun to go through this through this argument. In fact, it requires a little sip of water here. Sorry. So why do we call it? I like to call it money. You can also call it a rechargeable battery, or a well, there's some other things. But anyway, it's just sort of maybe a silly argument here, but we as humans, we use our wits and energy to work, and we generate money. In other words, if we want a car, we don't go to our employer and say, I need a car. So he says, well, you can work for 6 months and then I'll buy you a car. We actually use money as a means of exchange. We work, we get money. When we want to buy things, we use that money to buy things. So there's really no connection between our employer and our spending. Money is the intermediary. It's been that way for a long time. Humans invented that, maybe not billions of years ago, but at least a few thousand years ago. So our cells, by analogy, our cells work, process food, and they generate ATP. And then our bodies use the ATP to do stuff. So it functions like an intermediary between processing the glucose and using the energy that glucose gives us. I'm going to talk more about this stuff here, but in fact, the ATP that we generate from our mitochondria, we use it basically in 3 ways, and approximately 30% for each, depending on how active we are and so on. To move, we walk around, our heart pumping all the time, so it requires ATP constantly. We think, our nervous system requires an enormous amount of ATP, and I'll explain why that is. And we build things. We build new cells, we repair cells. We have to build things constantly. And that requires about 30% of our ATP. So again, here is the ATP. This is sort of redundant from what I said before, but when we use it, it loses a phosphate. Here's the ATP that we made in the mitochondria. When we use it, it loses a phosphate, so it no longer has energy left. We expend the energy here. Now we recycle the ADP to make ATP. I just pointed out how we can consider it to be money. We can also consider it to be a rechargeable battery, or a credit card that we sort of recharge all the time. But anyway, it's an essential concept that we generate the energy, then we use it, but those things are actually separated by this reaction. So first of all, talking about movement, this is sort of a typical musculature of a 70-year-old man. This is what a muscle looks like. And if you, they're stringy. If you take a steak, I mean, we all know that if you take meat, and sort of tease it apart, you have fibers. So these fibers are called muscle fibers. And within a muscle fiber, you have even smaller things called myofibrils. And these myofibrils, if you tease them out with appropriate, you can sort of separate them from each other. Then you can look at them in a microscope and you see that they have these bands. So it's an interesting little, it's a cute molecule, a cute cell. Look at them, just drawing another picture. This is what we call a, well, this is again a blown up myofibril. And if we take this thing here and look at it, this is a set of 3 muscle cells together. These things here are called sarcomeres. Sarco refers to meat, and a mere is a unit. These are sarcomeres, and these are really the functional units within a muscle. And you have a whole muscle, but in fact, you don't have, it doesn't work as a whole thing. These are all little cells that by themselves function. They're made up of thick filaments, these things here, and thin filaments. And I'm sort of telling you these things because these are what I spent about 30 years working on. So muscle contraction, how in fact is ATP used for a muscle to contract? And again, I'm reminding you that the heart, every second, things are going back and forth, back and forth. This is an idealized picture then of a sarcomere. I showed you the dividing line between one end and the other, and it has 2 myofibrils. This one here comes in from this direction, and this comes from this direction. And they are in fact polarized. That is, one goes like this and one goes like this. They are they're not sort of parallel. They're opposite in direction. This thing in the middle is what's called the thick filament, and again it is also polarized, and it's polarized in this way. The myosin the molecules making up these things have a long tail and 2 heads, and they go anti-parallel, starting at the beginning. In the middle here, we have these long tails hooking together, but one set of molecules looks in this direction, one in this direction. So in fact, the way this faces this is identical with the way this faces this. When they contract, nothing gets shorter, nothing actually, it's not like a rubber band. These things slide past each other so that this part slides past this. And here, this slides in this direction. This slides in this direction. And so that relaxed, it's like this. Contracted, these are closer together. That's why a muscle expands. If you shorten your muscle, it gets fatter, but it doesn't, but there's nothing in there that stretches. So where is the ATP used? Well, this is the way muscle works. We can, again, everything in biology seems to be cyclic. We start here with a myosin head in what's called a high energy configuration. It's bound to an actin. These are actin molecules. These are molecules. These are actin molecules, these little blue things. This attaches to this, and ATP is attached there. ADP is attached there. Then it goes to this stage where the myosin head sort of, we call it relaxing. It's actually pulling this actin in this direction toward me. These things fall off, and the myosin head dissociates them from this and binds an ATP. The ATP is hydrolyzed, is broken to binds an ATP. It is hydrolyzed so that it becomes an ADP and a PI, and it's separate from the actin. Then when this flips back, it flips from here to pointing straight over here. When it does that, it binds again to the actin, and then it remains bound until this point here where the ADP falls off. Then it goes back to this shape, and it has pulled the actin toward me. Then it relaxes, releases. This goes back to its original shape, binds. Here it's bound. It pulls the actin here when this falls off, and it releases, and it just keeps going like that. To do one cycle takes about a millisecond. There are differences in rates of how fast muscles work. There are slow muscles. There are fast muscles. For example, the muscles in your eye that control eye movement are extremely fast. The muscles in your legs are fairly fast, but not the fastest. Then there are some slow ones elsewhere. But and these reflect slight differences in the molecular properties of actin and myosin. Well, just if I may just indulge you, I spent several years working on the structure of this molecule. Determined its amino acid sequence. It has 384 units, and I figured out how. I say with my technicians and students and so on, figured out that, and a large part of this molecule here. That was basically my 35 years of working in the lab. So that was muscle. Brains and nervous systems also use a lot of ATP. I'm not going to go into as much detail here. I'm just going to say that all animal cells have to maintain electrical potential across their membranes, across the outside membranes. So there's a whole field of electrophysiology that studies this. But basically the reason you have a potential, the way to maintain this potential is to have more sodium on the outside than potassium on the inside. And that will give you a potential across this membrane. And the way you establish that potential is there are these little gates, these little gaps in the membrane. And they can open and close. But there are ATP, there are transport molecules, transport enzymes that use ATP the energy from ATP to move these things out and to move these things here. When an action potential, when a nerve impulse goes down the length of a nerve or within your brain, what happens is that this membrane temporarily depolarizes. If potassium goes out, the sodium goes in, and then that is reestablished immediately. These things happen very, very fast, constantly, and it requires as much energy to do this in your nervous system as it does to run your muscles and heart and so on. And the third way we use ATP is to build things, proteins, including enzymes, tendons, cartilage, DNA, RNA, fats, other lipids, blood cells, the works. Everything required by a cell is built by the cell itself. The energy comes from ATP. Any questions at this point? I mean, this is, there are just a few more slides. Yes, sir. Yeah, there's a whole, the question is, what are the effect of steroids on muscles? My son, who's a former Army Ranger and works out constantly, has lots of muscles, looks about like that guy I showed. He's always asking me about, about nutritional effects on muscle and doesn't worry about steroids at this point. But yes, there are certainly lots of influences that are very complicated, and people know about this. I don't. Yes.
I thought about that aging process. One of the most frustrating things that as I get older, I wear out faster. And is that the loss of age? And what is exactly happening?
What happens when we age and don't have quite the energy and ability to pump as quite as much iron as we used to. It's a combination of things. Part of it is circulation. Part of it is, I guess, wearing down of cells. There's a whole field of research called geriatric research, I guess. Maybe the physicians here would know more about that. But things do wear out. I'm acutely aware of that this week because I turned 85 on Friday. And so I'm not quite as active as I used to be, although my golf scores are still reasonably good. But yes, we do slow down with age, but it's a combination of things. And people, and very different people age at different rates. And so some people at 60 are as worn out as others at 90. But I really can't elucidate that very much. Yes, sir.
I really want to ask, we know that we can split a water molecule with electricity, take X number of electron volts, split one water molecule. In the case of photosynthesis, where the water molecule is split, is there some catalyst so that less energy is required to split a molecule of water, or is it the same electron volts of energy, whether it's done biologically, by chlorophyll, or by us with electricity?
Yeah, so the question is, compare the generation of protons or hydrogens in photosynthesis with the sort of laboratory methods that we use to generate hydrogen, for example, in trying to generate hydrogen for hydrogen cars or other uses of hydrogen. And I obviously I don't know the answer to that question, but the unit that I showed you, that is that catalytic center, is certainly key to it. And knowing what the structure is has given researchers optimism that perhaps they can replicate that in the laboratory. But it does in fact function as a catalytic center to apparently water molecule binds in some way to that center, and the electron configuration of the molecule changes such that it falls apart. It requires, it's a catalysis. I guess that the energy actually is not, it's lower energy in the oxygen, protons, and electrons than in the water molecule itself. That's why you don't require energy to do that. You require the catalyst. I think that's good. And so that catalyst is the key. And it's nature is very well. It's tried out a lot of things, but I don't know if it's going to be possible to replicate that. But, I mean, knowing the crystal structure of the complex, that is the protein and the sort of cavity in which this prosthetic, we call it this reaction center, the manganese and calcium, we sort of know what that structure looks like. Now, whether that can be replicated in the laboratory, I don't know. I mean, people are attempting to do that, and that may be, some people hope, a way to generate hydrogen. But whether that will happen, I don't know. Okay. Doug?
Is human muscle work the same as
Yes. It does human muscle work the same as bullfrog? I didn't work on bullfrog muscle. I worked on bullfrog hemoglobin. Bullfrog hemoglobin is similar. I actually worked on hemoglobin from Arenicola, which is a worm that lives down in the mud around Woods Hole, Massachusetts, for a while. And nature has, things have been around for so long, and many things have been tried because, as I pointed out last week, the key to to the origin of life is that it's mutable. It has to have DNA. DNA dictates the structure of everything, but these chemical reactions are never complete. They're never perfect. And so mistakes are made. And these mistakes then are sometimes, they're tested. So you get a slightly different molecule, get slightly different hemoglobin. And if the individuals who have those mutant hemoglobins are more successful in their environment, then these things are kept. And that's sort of what we consider to be the basis of evolution. And over billions of years, almost everything could have been tried. And that's one reason why I think we have molecules that are still here after all this time, ATP synthase and molecules that process glucose tend to be conservative. They haven't changed all that much. And ATP synthase. It's just unbelievable that cyanobacteria that have been around for a couple of billion years have molecules that are almost exactly the same as we have. Profile. Yes.
My question is similar to Steve. Is sleep a little bit like paying off your credit card? Where you spent the money.
Well, if you don't, is sleep the same as credit card? Sort of, but if you dream, you're using a lot of energy for your brain.
Then I'll rephrase the question: is there a deficit and surplus of ATP in my head?
Well, obviously it's not absolutely constant throughout the day, but it's the question is, what about during a day? The needs and surpluses change, and I would just simply say that rebuilding stuff, making cells, goes on constantly, whether you're asleep or awake or whatever. Your gut is constantly sloughing off cells from the inside surface of it, and they're constantly being rebuilt. So it's estimated that a 70-year-old man, I wrote down some of these weird facts. It's estimated that a 70-year-old person, a youngster, has made approximately £4.5 million of ATP in his lifetime. I mean, these things are going constantly. The typical ATP ATP synthase is making 100 molecules per second in your body. It's just very, very busy place, your cells.
Just like the guy
So, so what do plants, just a couple of things here. We're 10 minutes away, and I think we're in pretty good shape. What do plants do with glucose? Well, they do respiration just like we do to take care of their bodies, and they use glucose. They make glucose. They use glucose. They also make starch. And why do they make starch? Well, generally, the biological reason is they're giving their seeds a source of energy for after they germinate. They need to grow and fill their bodies. They make cellulose. Why do they make cellulose? To support their own bodies. I mean, a tree is a lot of cellulose to maintain its structure and to protect their seeds. So cellulose versus starch. Think of them like this. In fact, I have I thought I should have a show and tell. I told you that there's alpha glucose and beta glucose, and that beta glucose, when it combines, makes a molecule that's difficult to break apart. And the example of that is that this is a double knot, so we can't really break it apart. It's pretty tight. But alphas, when they go together, make a knot that we can break easily. So that's like a shoe knot. So we do this and it falls apart. So there are these absolute differences between between these connections. And in cellulose, this is the structure of cellulose, and it simply shows that these connections are what we call beta connections. Again, they're just schematically here, but these are difficult to break. This is starch. These connections are easy to break. So what is the importance of that? Well, starch and cellulose themselves are chemically identical. They're both made out of glucose. Glucose is the same molecule, but they're alpha and beta, and they're held together differently. So with the result that we can wear cotton, but not potatoes. We can eat potatoes, but not cotton, okay? So here's potatoes. I grow potatoes. I have kind of a cute picture. And again, these potatoes, and here's a really cute picture of a potato plant. But these things are just full of starch. Wheat kernels, a lot of starch, 70% starch. They also have some protein. Cotton fibers, on the other hand, are 90% cellulose. I was surprised to see that number. But these are these fibers, and these are, I don't know if you've ever seen a cotton field. It's pretty impressive. But all these fibers are cellulose. Cellulose is the most abundant natural product on Earth. Wood, leaves, flowers, grass, weeds, whatever. It's very abundant. And finally, oil and natural gas. Phytoplankton, and I'm not an expert, I consulted with my resident geologist here and read some stuff about it. But phytoplankton, I talked about these last week. They just live and die and fall to the bottom of the ocean. And so for over hundreds of thousands of years, you just get a whole pile of dead phytoplankton. They made glucose, they released oxygen, but they died. And they settled to the bottom. Ooh. And they were eventually buried under, man, I'm forgetting how to use this. They were buried under by earthquakes or tectonic movement, and high pressures plus heat from the deep in the earth turn that material into kerogen, which turns into oil, natural gas, and so on. Requires hundreds of millions of years, but every molecule of that oil came from something that was living, and its body entity was made by photosynthesis. Same thing is true of coal. That's a more recent thing, but apparently during this period of time, the plants just, they got on land, they grew, they died in swamps. I guess the swamp behind our house ways, the Hudsonville muck was all one of those. They died in the swamps, largely anaerobic. Plant material did not really decompose very well. And so large amounts of this were eventually buried under rocks, compressed, often at high temperatures, lost a lot of their hydrogen and oxygen, became coal. Now we have various types of coal. When I was growing up on the farm, we actually had a Franklin stove in the kitchen that provided us with our heat until we got central heating after I left home. And my dad favored Pocahontas coal. Apparently it's still around. It's from Pennsylvania. It's not anthracite, but it's good hard coal. And some of the anthracite has carbon levels near 90%. But again, all of coal's carbon dioxide made by photosynthesis. So once our first little prokaryotes, these were the first little animals that were there, billions of years ago, with DNA, whose intrinsic chemical properties inevitably led to slight changes with each generation, natural selection was certain to lead to new species. These changes and then survival of the species that were able to live and reproduce, and it's important to always remember that It's survival of the fittest, but in that particular environment. So it gives Earth millions of species living in every conceivable circumstance. I mean, in the hot springs of Yellowstone, or our skin, the floor, there's living things everywhere, and they all find their niche. Photosynthesis, using photons from the sun, gave living things an essentially inexhaustible supply of energy. Stephen Jay Gould, who's been gone for many years now, once posed the unanswerable question: if we could roll back the movie back to the point at which life on Earth began, let it run again, would the same plants and animals have appeared on Earth? That is, would we be here? We'll never know, of course, but overall, we can perhaps agree that things worked out reasonably well. Oxygenic photosynthesis was the key. Thank you very much.
Are there any questions? I have one. You showed us an image of photosystem 2 and an enzyme in which were embedded 4 manganese atoms. There are a few examples of that where an unusual atom is embedded. I'm thinking of vitamin B12, which has cobalt in its center. Are there other examples in the photosystems of the use of unusual atoms in their systems?
The question is, are there other examples of other atoms besides, say, manganese and calcium, as is found in photosynthesis, photosystem 1, other examples of other things in, for example, in making photosynthesis, or even perhaps in other systems? And I think that the answer is no. That is, this photoreacting center, this oxygen evolving center, is absolutely unique in nature. That is, this protein that contains 4 manganese and a calcium in that particular configuration, permitting the release of oxygen, seems to be unique.
You showed the nerve system, how it works and everything. There's sodium, potassium that are in there. We know that they are a positive valence and they want to combine with things. And I imagine it's a whole separate class to tell us how that's comes to be, how it's broken down from whatever it's bonded to, and how it ends up in our nerve cells.
The question is, what about sodium and potassium? Well, actually, you asked that on the microphone. Yes, no, that's the field of electrophysiology, where I was doing graduate work. There were people who were, in fact, the chairman of the department was an electrophysiologist. But in fact, the ions are floating free. I mean, sodium and potassium really they don't need to combine with other things. The transport of them, it requires an enzyme, the transportase. And I'm not sure what the status is on studying of those structure of those enzymes. That's the usual way biologists go about this. Try to figure out the structure of something, and then understand how it works functionally. You really can't understand its function without structure. That's why biology back in the 50s, when I started by talking about Philip Crook, who had taught physiology at Hope College and really piqued my interest in research. Back then, we knew nothing about the structure of these enzymes. We knew that they function. You could sort of isolate them, but we had no way to know what their structure was. But the structure is so key. It's key to function.
Yeah, just getting back to your question, maybe I misunderstood the question, but there are other molecules like copper, for example, in the molecule ceruloplasmin in the body that's involved in the immune system. And there's Wilson's disease, which is an abnormality of the copper metabolism that you can actually pick up in the iris of the eye. Anyway, and there may be other examples.
Good point. No, absolutely.
Indeed, there's a whole field of organometallic enzymology out there. It's fascinating. Any other questions? Very well. Let's thank our speaker, Marshall Elzinga. Thank you, Marshall.